Muscles are bundles of fibrous tissue that contract to produce movement or maintain body position. Muscle tissue is made up of specialized contractile cells, also called fibers, muscle cells, or myocytes. Muscle tissue has four main properties: excitability, which is the ability to respond to stimuli; contractibility, the ability to contract; extensibility, the ability to be stretched without tearing; and elasticity, the ability to return to its normal shape. Through contraction, the muscular system performs important functions like producing force and movement, supporting the body, changing body posture, stabilizing joints, producing body heat, and providing form to the body. There are three types of muscle tissue: smooth muscles, cardiac muscles, and skeletal muscles. In a human body, skeletal muscles make up about 40%, while smooth and cardiac muscles account for about 10%. Smooth muscles are involuntary, non-striated, and found in the walls of hollow organs and tubular structures. They are innervated by the autonomic nervous system and help regulate diameter and propel substances through tracts. Cardiac muscle forms the muscular wall of the heart. These cells are unenucleated, striated, and joined by intercalated discs. They are innervated by the autonomic nervous system, with heart rate regulated intrinsically by the SA node. Skeletal muscles are voluntary, attached to bones via tendons, and used to move the skeleton. They are roughly cylindrical, striated, and come in various sizes and shapes, allowing them to function effectively over a wide range of tasks. A skeletal muscle is structurally organized into the muscle organ, fascicles, muscle fiber, myofibril, and sarcomere. A muscle consists of many fibers, covered by the epimysium beneath the fascia. The fleshy part is the belly. Connective tissue layers blend to form tendons or broad sheets called aponeuroses, which secure the muscle to bone. Within the muscle, cells are collected into separate bundles called fascicles. Each fascicle is covered by its own connective tissue sheath, known as the perimysium. Inside the fascicles are individual muscle cells, or myocytes, wrapped in the endomysium. These cylindrical muscle fibers are numerous, ranging from 10 to 80 micrometers in diameter, and extend nearly the entire length of the muscle. Each fiber is usually innervated by one nerve ending. The cell membrane is called the sarcolemma. The cytoplasm, or sarcoplasm, contains nuclei, myofibrils, mitochondria, and sarcoplasmic reticulum, among other components. Each muscle fiber contains hundreds to thousands of myofibrils. Myofibrils are composed of myosin and actin filaments, which are responsible for muscle contraction. A myofibril contains I bands, A bands, Z discs, M line, H zone, and sarcomeres. The partial interdigitation of myosin and actin causes alternate light and dark bands. I bands, or light bands, contain only actin. A bands, or dark bands, contain myosin and overlapping actin ends. Cross bridges on myosin interact with actin to cause contraction. Z discs delineate the lateral borders of sarcomeres, where actin filaments attach and extend. The H zone is a light area in the middle of the A band. The M line lies in the middle of the H zone and is formed by myosin-binding proteins. A sarcomere is the portion of the myofibril between two successive Z discs. The sarcoplasm, the fluid between myofibrils, contains key ions and mitochondria that supply ATP for muscle contraction. The sarcotubular system includes the sarcoplasmic reticulum and T-tubules. The sarcoplasmic reticulum regulates calcium storage, release, and reuptake for muscle contraction. T-tubules rapidly transmit action potential impulses from the sarcocona to the myofibril. The sarcomere is the basic functional contractile unit of the myofibril, lying between two successive Z discs. It is composed of two main active protein filaments responsible for contraction: actin and myosin. The sarcomere is where the sliding filament contraction occurs. During this process, myosin myofilaments ratchet over actin myofilaments, causing the sarcomere to contract. The I and H bands compress and expand, but the myofilaments themselves do not change size. The two main protein filaments, actin and myosin, are the contractile proteins responsible for muscular contraction within each sarcomere. Actin filaments are thin, extending from the Z line across the I band and into the A band. They are polymers of G-actin, forming a double helix. Each F-actin molecule has an active site for myosin head attachment. Myosin filaments are thick and situated in the A band. Each filament is composed of about 200 myosin molecules, each having a head and a tail. The tail is made of two heavy chains in a double helix. The heavy chains form a globular head portion at one end of the double helix. Each myosin head has two attachment sites, one for the actin filament and one for the ATP molecule. The head is absent in the central H zone. Regulatory proteins include troponin and tropomyosin. Troponin has three subunits: troponin I, T, and C. Tropomyosin molecules are situated along the actin filament and cover the active sites on F-actin in a relaxed state. Other stabilizing proteins include actinin, which attaches actin to the Z-line, desmin, which binds the Z-line to the sarcolemma, and nebulin, which runs parallel to actin filaments. Titin is a large, springy protein connecting the M-line and Z-line. It forms a network for the sarcomere, providing elasticity to the muscle.